So then the radioactive protein hijacked the bacteria cell, and it replicated. Just like coronavirus replicates in our body and our cells, it hijacks our things and it produces copies of itself. And what they found was that the next generation were no longer radioactive. So the radioactive protein on the outside didn't produce radioactive next generation viruses. They repeated the experiment, but this time they made the genetic information on the inside, DNA, they made that radioactive. And then they let the same process happen, and what they found, and I know it's off the screen, but what they found is that generation, the next generation, the one after that, they were still radioactive. So the DNA that was radioactive was the thing that got replicated and got passed on. So the conclusion that Hershey and Chase came up with is that DNA is the thing that leads to traits being passed on. So important things that DNA needs to be able to do, it needs to carry information from one generation to the next, it needs to be able to be replicated, and it needs to be able to determine these characteristics. Heritable, things that can get inherited or passed on from one generation. So we now know this, and it's like, oh yeah, we got our DNA from mom and dad. Again, it wasn't a thing 70 years ago. We didn't know what. I knew I looked like my grandma and my grandpa, but I didn't know why I looked like my grandma and grandpa 70 years ago, if I were back then. We got some stuff from my grandma, I got some stuff from my grandpa. We just didn't know what was the actual mechanism that passed from one generation to the next. In the end, it's DNA. And we, as science students, know that we get 23 chromosomes from my mom and we get 23 from my dad. And what are those chromosomes made of? And the DNA in a single cell in your body, if you were to go end to end to end, and like put it end to end and stretch it out, would be about 6 feet long. And that's in a single cell. So it's really, really small, it's really, really coiled, it's really, really compact, but there's a lot of information there. So to go through and say, hey, let's sequence it, it's a lot. And there's a lot of DNA out there in your body that doesn't actually code for anything. It's just there. But there's some that does and is important. So when we talk about parts of DNA, we talked about it already a little bit. There's this thing called a nucleotide, and I expect you to know what a nucleotide is. And it consists of 3 parts: a base, an A, a T, a C, or a G. A sugar, specifically DNA, is made of a sugar called deoxyribose, and a phosphate. So on that activity yesterday, it asked you to circle a nucleotide. A nucleotide consists of 1 phosphate, 1 sugar, and 1 base. That is a nucleotide. Sometimes people put 2 phosphates. Nope, there's only 1 phosphate. That would be a different nucleotide. Contains 1 sugar, 1 phosphate, 1 base. Okay? And it's only 1 of them. And the bases are A's, T's, C's, and G's. And again, if you remember letters, great. If you want to memorize adenine, guanine, cytosine, thymine, fine, but not necessary. There's a thing called complementary base pairing, and we'll talk about a different name for that in a moment. And that's this idea that they form complements, and A's and T's are complements of one another, and C's and G's are complements of one another, and that there's a certain relationship between those, and it's called complementary base pairing, or Chargos Rule, and we'll get to that in a moment. So if we think of DNA as a structure, if we think of DNA as a ladder, here's our ladder. The sides, the things that I grab if I was going to climb this ladder, are referred to the sugar-phosphate backbone, because it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. They form the backbone, or the main structure of that DNA molecule. And the parts we step on, the rungs of our ladder, are the nitrogenous base, or the nitrogenous base, or the nitrogen-containing bases. The A's, the T's, the C's, and the G's. Now there's these dashed lines. Those dashed lines represent hydrogen bonds. And hydrogen bonds are relatively weak bonds. But if you notice, between the A's and the T's, there's always 2 lines. Between the C's and the G's, there's always 3 lines. So that's important in terms, and I don't care about the type, the ring structures, just for the record, there's these things called purines and proteins. You know, you think of these as kind of like these hydrogen bonds as having a charge, because they do. There's positive and negative charges. So imagine if instead they were like magnets, right? So on this, I don't even know what this one would be, on this base, there's a north pole and a south pole. Well, the complement is going to be attracted, opposites attract, right? So the complement is going to be south-north. And they're going to fit in, and there's going to be two magnets that go click, and they match together. On a different base pair, it might be north-north-south. The complement of north-north-south is south-south-north. So north-south is not going to attract a south-south-north because it's not going to fit, right? 2 and 3 magnets is like, where does it attract? And it doesn't fit. So those hydrogen bonds kind of help the attractiveness and kind of that complementary base pairing. So the A's match with T's and the C's go with G's. An apple in the tree, cars in the garage. And that complementary base rule that your packet yesterday referred to it as complementary base pairing, also is referred to as Chargos Rule or Chargos. 1950, he did an experiment, and basically they took a DNA sample and they just chopped it up into tiny little pieces. And then they did something, I don't know what they did, but they were able to identify how many A's and how many T's and how many C's and how many G's there were in this bowl of chopped up DNA. And then he looked at it, he studied the data, and what he found was that the number of A's in that chopped up bowl of DNA appeared to be in the same amount as the T's, and the same thing with the C's and the G's. They were almost equal. Okay, now why weren't they equal? Because we're talking like thousands, millions, so they were pretty darn close kind of thing. So he came up with this complementary base rule, also referred to as Chargos Rule, that says for every A there's a T, for every C there's a G. Apples in the tree, cars in the garage. That's the memory device that Amoeba Sisters suggest. I often say for every C there's a G, for every G there's a C, for every A there's a T, for every T there's an A. I also sometimes look at the actual structure of the letters that we use. So a capital uppercase A has straight lines and only straight lines. And that capital A with straight lines and only straight lines pairs with a big T which has got straight lines and only straight lines. So the straight lined letters match up. C has a curve, G has a curved line. Those two match up together. It's just another memory tool. Pick one that works and use it. Apples in the tree, cars in the garage. For every A there's a T, for every C there's a G. Complementary base pairing or Chargos Rule. So 1950. 1950s, I don't know, I guess when she actually posted her project, there's a female scientist by the name of Rosalind Franklin. I'm referencing specifically that she was a female scientist because in the 1950s, females didn't get a lot of credit for their scientific work. They were often seen as unequals in the scientific community, and it was often like, well, they're working with Dr. So-and-so, so it's really Dr. So-and-so's work. They're just kind of an assistant, which is complete bogus. She was the one who did this, and she didn't really get her due credit until she was dead. So in the 1950s, she took this picture, the picture on the right, she's using a technique called X-ray diffraction, and it kind of helped plant the seeds in like, hey, this is what DNA really looks like. And she decided, well, it must be twisted in some way, shape, and form. And basically what it is she ended up taking a picture of the DNA kind of in this orientation. So it was kind of from a top-down view. Okay? And she's like, well, it must be twisted somehow. So she did work with X-rays, and unfortunately a lot of scientists that worked in X-rays at the time didn't realize the potential harm of being exposed to a lot of X-rays. And it's actually due to the work that she did with X-rays that ultimately killed her. She died at age 37 of ovarian cancer, which being exposed to a lot of radiation along the way is probably what caused it. But it's not a unique story of scientists in those early days of that working with that type of material that died of some form of cancer that probably was the cause, but they didn't know of it. So she died at the age of 37, and her work was pretty pivotal in these two guys coming up with what we call now the double helix model. So the two scientists, these are probably the two most famous scientists when we talk about DNA. They're the most recognizable names. Names are Watson and Crick. James Watson and Christopher, but often they just go by Watson and Crick. So they were the two that kind of came up with this 3D model, and there's a picture of the 3D model kind of there that you can see. And what they did is they saw Rosalind Franklin's work, and that she said, well, it must be twisted somehow. And then using her work, which a lot of scientists do, they say, well, hey, yeah, look what that person came up with. That's good, but I think they get a little bit better of an explanation. It's part of science. But what they did is they used her work to realize that, well, yeah, it's twisted somehow, but it's probably it's a double helix. So if we look at, here's their model, they have a whole bunch of like rings or support stands right here. And that idea is kind of like the spiral staircase, right? This is that center pole of a spiral staircase. And then these are the steps of the ladder, or our nitrogenous bases that are rotating around that fixed point. That's that idea of a double helix. They won a Nobel Prize in 1962 for their work to propose that DNA was a double helix, and they never gave Rosalind Franklin any credit. They didn't acknowledge the work that she did help them lead to their kind of findings in the model. So I don't know if this is a perfect analogy, but it's kind of like, and maybe this is painting them in a bad image, but it's kind of like an interview with an athlete, like a world-class athlete. And be like, hey, tell us about the game. Oh, I did awesome. Like, look at all, I scored a lot. We wanted to win, so they gave me the ball. Kind of like that, right? Compared to someone like, hey, tell me about the game today. Oh, yeah, today went well for me, but because of the work of the other people that I was working with. Because so-and-so had a great game, they were able to really, like, free up some pressure on blah, 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 insert the, or looking at Academy Award speech for Best Actor, Best Actress. They don't say, hey, thanks, yeah, I was pretty good, as some people do. But the idea is, hey, they realize that where the award they're receiving, Nobel Prize, pretty prestigious in science, was because of, hey, thank you for this award, but if it wasn't for the supporting actors and actresses that I worked with, or the production crew, or the director, or blah, 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 there's a lot that goes into these, and Vegas kind of never gave her credit for the contribution. So because of their work and the works of the people that preceded them, in terms of DNA, we now have a pretty good understanding of the shape of it, and we refer to it as a double helix model of DNA. So it looks like a twisted ladder or a spiraling staircase. There's these things called hydrogen bonds that hold these base pairs together. And that's kind of my magnet analogy, right? There's magnets, there's an attractive force between these things that are holding them together. They're kind of strong, but not super strong. They're strong enough to hold the base pairs together, but they're not so strong that they can't be broken when needed. And it is needed. We do need to break those hydrogen bonds when we replicate DNA. We need to break those hydrogen bonds when we make mRNA to make proteins. So they serve their purpose. They hold them tight when they need to, but they're relatively easy to break. And the sequencing of that is there's these hydrogen bonds that are held together. Enzyme called helicase comes in and it breaks the bonds. It's like scissors. It breaks the bonds. Then that molecule is pulled apart. Then these free nucleotides are attracted. There's these chunks of like nucleotides floating around. And they say, oh, there's one, and click. They come and click in, and a new bond, hydrogen bond, is formed. And that's part of the replication process. So we got to cut the bond first. We attract the opposite, A's and T's, C's and G's, the complement. Then we form a new bond to make our replicated DNA. Okay? So, those are some structural things with hydrogen or with DNA. Yesterday you got that activity that if you didn't finish, you should finish. It's due in Canvas tonight at 11:59 p.m. If you did finish, awesome, great, make sure you submit it. I hear too many stories of people like, oh, I had it done, I just forgot. Now, one of two things, don't lie to me. Did you really have it done? Which, sometimes, absolutely, they did have it done. I forgot. I get busy. I forget things too. But secondly, if it's done, turn it in. Get it in. Take care of it, okay? So let's not have those little silly mistakes along the way. So your POGIL activity, there's 5 pages. Make sure you get those in and submitted into Canvas. Due in Canvas by 11:59 p.m. tonight. You have some time, about 10 more minutes here in class to wrap that up. And any questions you have still, I am available to answer questions for that.